Okay, so let's get started this morning. Hope you all had a good evening last night at the quiz. If you were out there, hope you got as many questions right as we did, which was some. It was very, very hard. I was surprised at how little I realized I knew. For those who don't know me, my name is Matt Williams. I am the president of the Society of Research Software Engineering. And it's my pleasure to introduce our platinum sponsor for the conference and our keynote speaker this morning. So before we start talking about the speaker, I just want to say a few words about the platinum sponsor, CloudKubed, and how we at the Society have been working with them and trying to form a collaboration on an ongoing basis. So CloudKubed's been working with universities and government and research institutes for a long time now in things around secure data, data transfer, data management, that kind of thing. And they approached us at the Society seeing that the kind of model that we have as RSEs, the way that we share our knowledge and expertise, the way we have pools of experts who can move out into different teams in a flexible, dynamic way was really appealing to the kind of work that they were doing, working with universities and working with the private sector in CloudKubed. And so in our work that we're sort of trying to do along with CloudKubed, there's sort of two main themes that I'm seeing are coming together that are being satisfied by this collaboration. First of all, we're trying to form some longer-term partnerships with organizations outside of just our society. And this is potentially going to include universities, companies, private sector, funding agencies, whatever, to try and break outside of this mold of just having annual sponsorships for the conference, where we have these short-term partnerships, and then by the time the conference is over, we just hope that it's all been okay, and then the next year we have to do it again. We want to form long-term partnerships with companies and groups who really understand what RSE is about, what it can do for them, and what they can do for us. And so CloudKubes are really invested in that and really keen to be involved in that whole process. And on the other side, there's long been a history of RSEs, especially at this conference and the community that you see, being very university-focused, some research institutes, but RSEs who are working in industry, out there in companies, in government, places like that, it hasn't been as much talk about them. And I've been really heartened actually this year at the conference to see more and more companies coming along saying we're doing RSE, we're part of this community, and you are very welcome, and we need to make sure we carry on working in that way. And so we're working with companies like CloudKubed. We're hoping to sort of soften that barrier that you get between academia and industry, and allow RSEs to move back and forth between the two, sharing knowledge in both directions, making sure that we all understand and learn as much as we possibly can. So if anyone wants to go and talk to CloudKubed, they've got a stall out there in the main atrium. Who here is from CloudKubed? Could you wave your hand? We've got a few down here in the front row. Thank you very much. And of course, we've got our keynote speaker, who I will now introduce. So Manjinder Sandhu is a professor of population health and data sciences at Imperial College London. He trained at King's College London, at the London School of Hygiene and Tropical Medicine, and the University of Cambridge, and his research focuses on the interpretation of large-scale health and genomic data, and integrating technological advances at the population health level. His work at CloudKubed is focused on advancing research potential and solving society's toughest challenges through innovative technological solutions, including high-performance computing and trusted research environments. Please join me in welcoming Manj to the stage.
Thank you. Just making sure this works, and yes. Thanks for the introduction. I just want to start off just by giving some of my headline views on what I hope you glean from the talk today. So fundamentally, much of what we've understood in the context of discovery science has largely been the result of collaboration and sharing information. And I'm a massive and firm believer that sharing information and data with as many people with the right attitude and the right mindset and the right motivation to understand and solve the problem will give the greatest benefits to human population health and I think society more generally. And I don't use the words bright people, I don't use the words clever people, I use the words attitude, motivation, to answer and solve the problem in front of them, because I think those are the two most important attributes and traits that I've observed over the last few years about doing large-scale science in discovery and the collaborations we've had. People are just generally interested in solving the problem. They have enjoyment in trying to solve the problem, and those always end up giving you the best results. And I'm a fundamental believer that those traits are the key path to success in solving the problem before you. I think another aspect of what I've learned over the last few years is to think about critical articulation of the problem you're trying to solve. And I think over the years, it's become more and more entrenched in trying to understand that and evaluate that really critically. Sometimes we want to do stuff because we're fundamentally interested, but it may not necessarily solve the problem in front of you. And I think trying to disassociate some of those things as part of that journey is really important for you to execute and deliver, and ultimately that's what we try to do. And in academia, there's a really clear benchmark. You think of an idea, you do some science, you communicate that within the context of a research publication, and you get it published. And you go through the pain and angst of peer review. I'm sure it's lovely for some people. But ultimately, there's a clear, hard outcome of your work. That naturally translates into this idea that amongst your peers, that we can trust you with funding to continue that endeavor. And the cycle continues. Some people may find that crazy, but fundamentally, that's the business of what we do in academia. And translating most of that into the commercial context is really to start thinking about how we can even expedite and enable and innovate to a greater degree. And for me, the motivation of working in the industry construct is largely around that. Can I take more risks? Can I innovate more? And can I develop infrastructure, resources, and science in an unprecedented way? And that really, for me, is the motivation for straddling both academia and industry to really maximize opportunities to deliver the work we want to do. I thought I'd start and just give people a sense of how I came about the idea of moving into population health and translating into thinking about large-scale data science as a framework for the work we do. And originally, when I was intrinsically interested in population health data, not individuals, but the population scale level data, one of the questions we used to consider and ask ourselves was, how do we take information we glean observationally and create statistical models to understand how the world around us works, to look at more causal frameworks for those observations? And the reason for that is, once I fundamentally understand the cause and drivers of disease or some artifact or manifestation in society, I can start to think about interventions. And as I worked within population health, the key element that started to attract me was to think about how I could align genomics, because the intrinsic properties of human genomes, how we inherit them, and the inferences we can make from them might help me get closer to the biological causes of diseases, which ultimately might help us either reposition drugs or create new medicines for people. And that, for me, was the big driver to start understanding, one, what is genetics, what is genomics, what is the genome, what is the patterns and correlative structures in a genome, and how do I use that to understand something about populations and its impact on future disease risk. And the way we wanted to answer some of those questions was not to be opportunistic. So you imagine a world 20 years ago where there were lots of genetic associations linked to many, many diseases. Whether they were true, whether we could have validity or reproducibility was a major critical limitation in our understanding. As we started to build technologies to sequence genomes, to start characterizing the human genome, to start understanding some of the patterns of those genomes, we could start to build technologies that allowed us to start characterizing humans at greater and greater scale. So rather than just think about a few individuals where you can characterize some of their genetic information, can we move to thousands of individuals? That started to give us an idea that with this information, we can statistically resolve or reproduce or attempt to reproduce some of the earlier observations. And we realized that if we can do that as a collective, we would have much more statistical resolution. So the interesting thing about genomics and population scale genomics is that they were probably one of the first groups globally to really understand this idea of collaborative work. if you're doing Project X and we're doing Project Y, can we come together because we can statistically resolve the observations to a much greater degree? And as we've started to more densely characterize the genome, we realized we're now looking at millions and millions of variations in a human genome that we are now testing for thousands of clinical phenotypes. That creates a statistical problem that we also need to resolve. So it increased the imperative for us to come together in large-scale consortia of hundreds of thousands of individuals within population data sets, millions and millions of data points, to start to think about how we can learn from that information and make more definitive statements about the associations we observe, which could lead us into this idea of discovery science. So importantly, we would not have got to where we have, and now there's thousands of discoveries. The next transition in genomics is to think about how we think about validation of those and target validation of that information. But equally, we're continuing to scale. Even to this day, the mandate for most industry, pharma, biotech, academia is to understand the variation in millions of people. And that is happening now. And very soon, it will reach 10 million people that we will start analyzing data on, with each of them with probably 50 million data points. You can imagine that information is going to scale and scale and scale, and we'll need a framework to deal with it, both in terms of how we even generate the information. Do we even keep image files of genomes? No, just throw them away once we get to the VCF state. So you start to make different decisions because you have different problems to solve as a function of the scale of data you are now producing. As we thought about this problem of large-scale data, one of the early parts of my thinking was to consider how we would capitalize on global human diversity to improve our ability to discover genomically. So you imagine most of the work has been done in the context of the US or the UK or other high-income countries that had the resources and the funding to start thinking about how we could do large-scale genomics. Most low and middle-income countries were not participating in genomic research. But one of the key things we identified quite early on is that when populations segregate over long, long periods, the mutations they accumulate, because we accumulate largely mutations because of random drift, they're just random mutations, there's new opportunities to look at the genetic structure of different populations that have been continentally isolated relative to each other, where we could start to see how we could better refine our genetic association signals. So you imagine, for those who are not that familiar with genomes, you imagine if you have mutations in a genome that are closely geographically located, they tend to be correlated. And so it's hard to disentangle the causality of one relative to another, because I don't really find individuals that can reliably have those on what might be called haplotypes, different strands for the individual. And so what you want to look for is populations that have a different pattern of correlation around the world, so you can start to statistically resolve some of those signals and find ways to better refine. And so looking globally, the decision we made quite early on was to think about, well, how do we capture variation in human populations at scale? And that really led me to the idea of thinking, well, why don't we start to characterize human populations? There was the 1,000 Genomes Project that started at that time. There was the Human Genome Diversity Panel, which looked to characterize from a population history perspective. And so we started to think about how we could identify and characterize genetic variation in different population groups. And that really led me into another field, which is population history. So the argument we made in this context was to build genomic resources that could be utilized for medical genetics. The first thing I probably need to understand is what is the makeup of that genetic diversity, and what is the source of that genetic diversity? Because the more information I can collect that recapitulates that information, the more I can build new methods or new resources that can start to support those studies going forward. So part of that meant, let's characterize diverse populations. Let's understand some of those patterns of human movement, migration, admixture, and there's really good models to start understanding some of those interactions. And we can make statements about population history. And here, this work we did, which was a paper published in Nature a few years ago about medical genomics and how we could create resources. What this really starts to tell you is that as we see population movements thousands of years ago, we can start plotting admixture relationships of different population groups, which can tell us something about population movement. When segregating populations mix, as I told you, that most of that variation is random. But if you mix, you'll start to see correlation in some of the differences between mutation frequencies between population groups. And those mutations and those correlations will have a pattern and a curve. And it's that curve you can use through, and using simulations, you can start to understand the dates in which those admixture patterns may happen. And that's just taking a suite of metrics from a totally different field. It's called a suite of F statistics that we use to start understanding something about population history, time points of admixture, which starts to tell you something about the composite makeup of that genome, which means if I can increase representation of those genomes, then I can start to build new tools for medical genetics. And that really was the kind of natural path for us to move forward in the context of the research we did. I've reiterated this idea about collaboration. And for us, it is all about collaboration and sharing data, which has implications for how you might think about research software engineering. How do I share information reliably, sensibly, securely with different researchers to come to this idea of building a collective resource? Part of what we wanted to do within the context of building research within communities was making decisions about how we support capacity in the global population groups we studied. And we had two modalities. Lots of people were thinking about genomics and saying, hey, you know what, we should just build a sequencing facility because we should generate data. And our argument, working with institutions within Africa in this context, was to say, it's really hard to sustain a capital-intensive endeavor like sequencing in the long term. And we can see organizations in the US and the UK struggling with those endeavors in the long term. How do you support a highly capital-intensive service like sequencing genomes? And our conclusion was that actually to create ownership and sovereignty of data, we should enable the analysis of data. And so we took a large amount of money from the UK government and built a data center in Uganda. And this was a group of us at the Sanger Institute at the time, a very interesting way to build a data center, put it on some pallets, set it up, fire it up, it works, turn it off, ship it to Uganda. And we were lucky enough that the Wellcome Trust said, hey, we've got a building. Let's put a building together. Okay, let's have that building. Let's put a data center in the basement of that building. We couldn't have the first floor. I mean, logically, we'd want it in another place. Fireproof it, waterproof it. How do I get networking to this place? Just so happened that the UK government at the same time was thinking, we haven't changed the electricity supplies and chain since the 1950s. So they started digging trenches. Oh, by the way, whilst you're digging those trenches, could you put these cables through as well? Which they did up to the gate. We finally had a conversation with Google, and that Google at the same time was saying, we're trying to get fiber to everybody because we want everyone on the internet. Oh, by the way, can you dig up to the gate? So they dug up to the gate, and we connected up to the Ubuntu Alliance network across Uganda, which links it to the rest of Africa. And you know, my greatest achievement in building this data center, which probably had 20% of the compute at the Sanger Institute at that time and still does, is that I have nothing to do with it anymore. That it's completely supported by the Ugandan Virus Research Institute, and they use that data center to support their activities and build that through sustainable grant funding. But what it did do was allow us to build another project around building one of the largest genome resources in Africa at the time, the Ugandan genome resource, which was collated with other data sets. And you imagine a computationally intensive data set. We needed to call those genomes. We need to merge, recall those genomes with other data sets. It was a massive endeavor to put that data together, but also link multiple collaborators around the world so we can analyze substantive sets of questions around population health within the African context, going from population history and diversity to understanding patterns of heritability given different environmental contributions, to understanding new and discovering new markers for genetic association, as well as the broad population history and substructure within local populations in the Ugandan community. So it gives you a sense of how you collaborate, how we might build resources, and the importance of creating frameworks for sharing data and allowing many organizations to use that information. And as you build those resources and give those to the community, anyone can access the Ugandan genome resource, you start to have opportunities to really capitalize on new discoveries. And this slide just captures some of the work we did last year, published in Nature, which talks about a completely new discovery for the biological regulation in human populations of HIV, a major problem within sub-Saharan Africa. Up to that point, most studies understanding the genomics of HIV regulation in human populations has been done in European populations. So this was the first large-scale resource to take information from a raft of populations within sub-Saharan Africa, working with a range of collaborators both across the region but equally globally, and to start to look at genetic associations. And here you can see that we discovered a completely new locus on chromosome 1. The biology was completely unknown in the context of HIV. When you start to look at that information and the genes associated with it, you can see that there's a clear opportunity and known paradigm for the biology of HIV in terms of HIV replication in human populations. If you don't know, HIV integrates with the human genome, and it's a retrovirus, and it uses that as the mechanism in which it will replicate. Here we found a molecule that increased expression of dampens the ability of the virus to regulate. And there's other work that we've started to build around, which is building synthetic RNAs at scale across genomes and using machine learning tools to predict how we can start to find new molecules and drug targets for this candidate as well. But again, it gives you a sense of, we've built a resource, we shared it with the community, that led to an opportunity where we could start to work with another group of global partners to create a new framework for HIV biology and bring in organizations globally to start analyzing those data. Remember, you have multiple skill sets. It's not a question of we do everything. It's a question of working out collaboratively together. How do you build the research software engineering tools to understand population history? How do you build the tools to understand genetic diversity? How do you build the tools to understand the discovery side of it in terms of genetic association studies? How do you curate and manage collectively phenotypic information that gives you all these insights? That is done through multiple groups thinking about specific problems that come together and coalesce into a discovery program. I probably want to end this talk just talking about how we're thinking about the problem more and more. I think when you work in an academic environment, it's really clear that in universities and the way universities are structured and the organic growth within a university, it's hard to control order and it's hard to create efficient systems for building infrastructure and analytical capabilities to deliver the science. Because everyone wants to do things in their own way, and the way the funding is structured makes it harder for you to really enable that. What we see in the context of how we see university organizations, how we see other research organizations really coalescing around this idea of using cloud infrastructure as well as on-premise physical data centers will be just a hybrid of the two. For now, I wouldn't argue that will be true in 10 years' time, but certainly over the next few years, we see a coalescence of both using technology that is best surfaced with the cloud versus that can be done within physical data centers where that capital expense has already been committed. And what we also see is opportunities to start linking those in a more efficient and prescribed way. How do you reliably create a framework that utilizes the best of your physical infrastructure, but also capitalizes on all the flexibility of compute within a cloud environment? And that's where we see the driver over the next few years for most organizations as they kind of transition and make key decisions about what they may want to do in the context of infrastructure through a hyperscaler versus infrastructure through their own organization. I think a key element that I also want to kind of talk about a bit is most discovery genomics in the context of machine learning, deep learning, AI, whatever the words are, there's already elements of most of those things, some of those things in the natural course of what you do in discovery science. In calling genomes, you'll use a calibration system, which is a machine learning tool. In estimating ancestry, we use a machine learning tool. We train a model and build that. I think increasingly, this falls to my first argument. Be really clear about the problem you're trying to solve and critically evaluate your solution for the problem. And then think about the complexity of what you're using to answer that problem. And that's the debate we always have in discovery science. Is it worth using this more complex, more black box tool when I get just as good as an answer using structured statistical models? I'm not saying that's true in all instances, but I'm saying it's always worth evaluating what is your quickest route to the answer, and are we creating complexity when we don't need to? And so again, I think the tenet for me is critically evaluate your solution, articulate your problem, and do it again and again and again, and then do it. And I think that to me has held me in good stead over the years, and I feel it's a good principle to at least consider as you follow in the endeavor. I'm going to leave it there. I think there was one last slide. Maybe we can circulate that afterwards. We've been working on some work around the Azure TRE and the accelerator program, and we're setting up a community group to kind of explore that in a bit more detail. We can surface some details of when those community forums are going to happen, but it'd be really great to at least see some of you there, and we can talk about these ideas of how we can integrate systems both in hyperscaling environments, but equally also how do we really get the best out of physical data infrastructure and systems. Thank you.
Thank you very much, Manj, for that really interesting overview. I think we're going to bring up some questions, but maybe I'll kick off whilst people are thinking of their questions and asking them on Slido. We often take data for granted as kind of research software engineers. We just see it as information. How hard is it to actually acquire particularly genomic samples? And when you're working in places like Africa, are there any kind of cultural considerations and stigma around that? And how important is it for us to think about that kind of upstream view in the work that we do?
So it's hard, and it's one of the hardest things to do to engage. We've worked with conservation refugees, indigenous communities, all the way to large-scale population groups, across the continent, Southeast Asia. And we always start with a process of how do we get ethical and regulatory approval, and how do we work with cognate institutions in those countries. And we start with that process in terms of adhering to legislation, thinking about the local population data laws, and how they impact how we use data. So one thing everyone should be really clear about, genomic data is personal special category data. It is not data that we can just use and shift and move from one place to another. So you imagine you have to think about the consent framework that was originally used for those data, how that data then needs to be handled and managed within the context of research, and that might mean special provisions within a data center, special access, registers of control, registers and records of processing. All those elements are a mandatory requirement in the UK. So you imagine we might build a data asset in Kenya. We need to follow Kenyan data protection laws. We need to follow EU and UK data protection laws because they're reciprocal in terms of how we manage information. If we share data with a third party, we have to sign an international data transfer agreement if we go out of the UK. We have to get a record of risk assessment as we move that data out to the UK, or even surface it through some infrastructure to a third party. And a good example is this. There are servers for imputation that are used. So imputation is basically the idea of predicting genetic variance in from very dense information to less dense information. And you imagine if I wanted to do that, I'm literally uploading genomic information to a third-party server in the US and then pressing some buttons. I need a legal agreement, I need an IDTA, and I need a risk assessment. That's what you have to do. If that data is collected under consent, you have to manage that in the context of the research environment you create, and there's no getting away from that. And the world is definitely changing, and that's why this idea of trusted research environments becomes paramount. You know, every organization should have one, at least some framework of managing data that is segregated to the least privilege. Why do you need to use it? And those natural things that need to be taken into account. So I think that has to be a key part of how you engineer your solution, because it will have impact on your analytical pipelines and just how we use data.
Thank you. So I have a question from Jack that's quite popular here. So it's great to see a data center gathering genomic data in Uganda being run by locals. Open data is important, but how do you ensure that the data generated can support careers of local researchers and not serve only to further the careers of established researchers in the global north
Completely.
and west?
And I'm going to talk about two antithetical things: open data and not open data. So what we do is we generate data, and we work with local partners to generate data. Do we make the data open immediately? No. Why? Because there are organizations in the world that will work faster and crank handle analyses and get all the credit for the work we've done with the local researchers than the local researchers. So the work we do is to control those data in environments that allow those researchers to maximize, and we support them to write those papers, and to enable their careers. So the key thing is to build that capacity. Why do we build a data center in Uganda? It's to enable science and create sovereignty within the context of Uganda. We're doing exactly the same in South Africa. We're building a trusted research environment at the Aurum Institute, an NGO, not-for-profit. That is an industry-led initiative that will hold the largest resource of genomics and healthcare data in the continent. But they will be analyzing that data. Local researchers will be analyzing that data. We work with tertiary referral hospitals in another context where we generate and integrate data on electronic health record data. And the idea, again, is to get local researchers to start analyzing those data, benefit from showing an organized, integrated data resource that allows them to do precision medicine clinical trials in the future. So you build capacity. And it's not quick, it's long, but the idea is to get hospitals to think about the data they have, the value it has to enable discovery science and translational clinical medicine, and to build the infrastructure such as clinical trials unit, precision medicine clinical trials unit, and that's the way you have to do it. And what that means is that you have to hand that all over at some point. And so it's very antithetical to some ideas of what you need to do, but the idea is to enable those communities by working with them, ensuring they are taking the scientific lead in those endeavors.
Thank you so much. To stay on time, we'll leave it there. If there's any further questions, please do go and speak to CloudKubed at the CloudKubed stand. And please join me in thanking Manj Sandhu.